Okay, so what we're going to talk about now is we are going to talk about um, how we get our sources of ATP, how we get the sources of ATP itself. So ATP, the actual content, the actual amount of ATP in muscles, it's relatively low. There's not much ATP just floating around inside of the muscle itself. So what this means is the muscle's got to find a way to actually generate ATP in order to sustain contractions, repeated contractions, because some of your muscles are under constant contractions all of the time, like your abs, your postural muscles on your back. Okay, they're always contracting, so they got to regenerate it. So we have three ways to do that. We have three ways to actually generate ATP. Okay, so the first one we have what's called creatine phosphate. All right, and this is an anaerobic process. And it gives you about 15 seconds of ATP energy. So using creatine phosphate, you can contract your muscles for about 15 seconds. So not the best, not the best. Okay. Then we have another way. It's called glycolysis. And those of you who've taken your Bio 101, you remember glycolysis is like the bane of Bio 101. It's terrible, but it's important because your body uses it quite regularly, and it's not very efficient because that lactic acid production just sucks. But anyways, glycolysis is anaerobic, just like creatine phosphate. So anaerobic just means there's no oxygen. You don't need oxygen to do it. However, the mechanism of glycolysis in the muscles can give you about a minute of ATP energy. So it's like four times more efficient than creatine phosphate. Right? The problem with this is we have those lactic acid byproducts, and that's bad news. Lactic acid byproducts just suck. And then we have the best one. We have aerobic respiration. Now, I'm not going to go into all this Krebs cycle stuff. You're going to get that other times if you haven't already done it. But one of the things I want to dispel that a lot of people say is aerobic respiration just won't break down sugar. Okay, aerobic respiration can break down fatty acids. It can break down pyruvate. It can break down the byproducts of glucose metabolism. It can break down nucleic acids. It can break down proteins. Okay, that's all of this stuff can do. So it can break down a lot of stuff. Okay, so just don't think glucose, but it's everything: glucose, fatty acids, proteins, etc. Okay, the thing here is you have to have oxygen in order to do it. Okay? You can't do it without oxygen. Okay? Now, most of the ATP that is generated by your body comes from this mechanism itself. And in fact, in muscles, about 95% of the ATP needed for resting or kind of like moderately exercising muscles. So, like walking, a good walk, the fat burn walk, that's where most of this is going to happen. And this type of respiration, this type of ATP production occurs in the mitochondria. Okay? So that's it. So those are the three mechanisms: creatine, phosphate. And where do we get that? We get that from creatine. And creatine, what it does, creatine phosphate is creatine, it's a protein, and you'll add a phosphate to it, and it makes creatine phosphate. And then if we need to make ATP, we just pull the phosphate off the creatine phosphate, and it'll make ATP that way. Again, not that much. Glycolysis, we break down glucose into 2-pyruvates, and we get 2 ATP that way. And then aerobic respiration, you need oxygen, you get a lot of ATP. It's the choice way to actually produce ATP in muscle itself. So moving on. All right, so now what I'm going to do here is, so a lot of students I've had, they've had this mental thing like how is the structure of actin and everything like that. So what I want to do is I want to actually I'm going to draw a couple of actins. Okay, so actin it's actually called F-actin, and it's a chain. It's a chain of protein. And on this actin, what we have, I want to draw them in green. At the bottom here, you have these things on each one of these little actin bubbles, call it like bubble tea or something. These are the myosin binding sites. Myosin binding. All right, that's what these are. Those are the myosin binding sites. Now, actin molecule is actually 3 molecules: F-actin, tropomyosin, and troponin. Now, tropomyosin, not troponin, tropomyosin. Draw it in this blue color. Tropomyosin is this protein that covers. It basically just covers up the myosin binding site. And that's where it sits. Sits right there, covering the myosin binding site. Okay, so we're going to say tropomyosin. Right there. Now, attached to the tropomyosin, you have another protein called troponin. Troponin. Right? Now, troponin's job, what it is to do, so we're going to do this. We're going to do this from the side. I'm going to draw it again. We're going to draw it from the side. So I'm going to draw 4 circles, but I won't label everything again. So we're just going to do that, and we'll draw a little arrow. Okay. So what we'll do is binding sites, and we'll show the binding sites like little circles. These are little binding sites. Kind of look like fish eggs right now. It's scary. Anyways, those are our binding sites. And then, no, binding sites are supposed to be green. Now we're right. Now I'm going to draw tropomyosin. Tropomyosin is blue. Now, this first one, we're going to call this one number 1. This is 1. Okay. This is pre-calcium. Right? So pre-calcium. That means calcium hasn't been released. So this means right here, tropomyosin, what it does is it covers these things. Tropomyosin covers them. So they are blocked. They cannot be used. Okay? And then on this, in this golden color, we have troponin. Troponin sits right there. Troponin has this little part called a calcium receptor. Okay, it's actually called the TNC. It's the troponin calcium binding site. So when calcium floats around, let's say here, calcium is going to be released. Let's say calcium gets released. And what calcium is going to do is it's going to float around inside of the cell, inside of the muscle cell, and it's going to bind in there. And what's going to happen when calcium actually binds in there is the tropomyosin, what it's going to do is it's going to move out of the way. Just like this. Move upwards. It's going to move. And what that's going to do is it's going to expose the myosin binding site. And troponin is still going to be attached to it. You still attach the troponin through the tropomyosin right here. It's still there. They're attached to it. However, attached to the troponin is a calcium ion. Calcium is inside of here. Calcium. I'm going to call this calcium. All right. So that's the arrangement. That's how actual actin and myosin are arranged. Sorry. That's how actin is arranged, and that is a relationship between actin, tropomyosin, and troponin. Okay. So literally, troponin moves tropomyosin to expose these binding sites. Okay? Now, if we were to look at it, we'll look at it from the side again. Let's just go from the side. And we're going to draw our binding sites again. Here's a binding site. Here's a binding site. Okay. And what this is going to allow is we'll draw it in purple. We'll draw the myosin in purple. What this is going to allow is it's going to allow the myosin head, that's what the head looks like, to bind to it. That's all That's all we got going on. There's nothing else. We'll do a lighter green color. And on the myosin head, there's an actin binding site. Now the actin binding site on myosin is never covered. Doesn't happen at all. It is the tropomyosin that actually prevents the binding to occur. So we have to kick that out of the way. And that's the importance of calcium. And that is the importance of tropomyosin and troponin, the regulatory proteins. Okay? So that's how that works. All right. Now we're going to talk about muscle relaxation. And muscle relaxation is really simple. I mean, it's really simple. Stop stimulating the muscle and the muscle will relax. I mean, that's pretty much all there is to it. When you stop stimulating the muscle, calcium will not be released. And if you prevent calcium from being released, just like I showed you, I can't bind actin and myosin together. And if I can't bind actin and myosin together, I can't contract. So that's it. So if I can't bind them together, I get no cross bridges. If I get no cross bridges, I get no contractions. And then the muscle will eventually just relax. I mean, it's really that simple. So don't make it difficult. How does a muscle relax? You stop stimulating the muscle. It is really that simple. Really that simple. All right. So cardiac muscle. All right. It's almost exactly like skeletal muscle, except there's a couple of little differences we're going to talk to. One, it's only in the heart. Okay. It's striated just like that. It's got sarcomeres just like skeletal muscle. The banding structure is the same as in skeletal muscle. Okay. However, the cells are shorter in skeletal muscles. They have typically only one nucleus, maybe two. Sometimes you'll see two nuclei in a cardiac cell. You will see this next semester when you look at cardiac muscle itself. The fibers in cardiac muscle branch, whereas in skeletal muscle, they're pretty straight. They're pretty linear. And they're all connected to each other by what's called an intercalated disc. Okay. And this intercalated disc is just a thickening of the sarcolemma, And in this, we have the gap junctions. And we have desmosomes. That's all we got. That's what you need about cardiac muscle for me. You're going to go in more depth with it next semester. And then we have smooth muscle. Now, where do you find smooth muscle? I like to think of this as visceral muscle. So it's around the organs. So that's where you're going to find it. In the walls of the organs, you're going to find it in the blood vessels, and you're going to find it in the eyeballs. Okay, so those smooth muscles, like to contract your iris and allow your lens to focus, those are smooth muscles. No striations. Okay, they do, but there's no striations in smooth muscle, but there is actin and myosin. It's just not arranged in a pattern that allows the light to be refracted strangely. Okay. Instead of being long lines, they're basically spindle shaped, which means they're wide in the middle and tapered at the end. So they're like spindles like this. That's what a spindle looks like. Okay. Single nucleus, pretty much like a cardiac cell. No sarcomere arrangement. They don't. However, the filaments, the actin, are bound to these things called dense bodies. Okay. And when the actin and myosin pull each other, it's going to pull the dense bodies closer to each other. So kind of like a Z-disc. So dense bodies can be like the smooth muscle Z-disc, if that helps you remember that kind of thing. Again, I'm not going to ask you questions like that. This is going to be next semester stuff. And the thing that regulates it, instead of troponin and tropomyosin, it's this thing called calmodulin. So calmodulin is what actually stimulates the calcium release. Calcium still has to be released in order for a smooth muscle to contract, just like a skeletal muscle. But that is it. So quick and dirty, went through muscles. Yeah, so just do the basics, get the basic stuff. Make sure you understand what calcium is. Make sure you know the difference between them, how a muscle, that a nerve has to go to a muscle. Know what the neuromuscular junction is. It's simple stuff. Don't try to be nitpicky and memorize everything, because life's too short for that, and it's really not important in the long run. Just know the basics, and you'll be pretty good for me. With that, we're done. Then congratulations if you listen to this a second time. Good job if you listen to your first. I will see you on Monday.